Welcome back, students. Molecular base of inheritance unit la we are going to see the process of translation in this video. So translation is also called as protein synthesis. Nama requirements for protein synthesis ellame paathitom. So we'll directly go into the process translation or the protein synthesis. The first step is going to be activation of amino acids. So amino acids வந்துட்டு டைரெக்டாக பார்ட்டிசிபேட் பண்ண முடியாது. ஸோ they have to first get activated. இப்போ இந்த activation of amino acids எங்கே இருக்கும் அப்படின்னா they are going to be present in the cytoplasm. So the amino acids present in cytoplasm வந்துட்டு they are going to get activated in presence of enzyme called amino acyl synthetase. So this enzyme is going to help in activation of amino acids. Amino acids eppadi activate aguna, we'll represent amino acids as AA. This amino acid in presence of ATP. So the activation requires ATP molecule for each amino acid, and in the presence of the enzyme it forms the reaction results in formation of an amino acyl complex. So amino acyl complex அப்படினா என்னன்னா amino acid bound to an AMP molecule, that is adenosine triphosphate வந்துட்டு monophosphate மாறி இருக்கு because there is inorganic phosphates that is released and the bond break ஆகிறதுனால யூஸ் ஆகிற எனர்ஜியை தான் வந்துட்டு இங்கே பைண்டிங்க்கு யூஸ் பண்ணுது. With this the enzyme is also going to be bound. So these three together, the amino acid, AMP molecule with the enzyme together is called as an amino acyl complex. So the activation is common for, so this process is common for all amino acids except methionine. This is because methionine can act as start codon, that is AUG, that is coding for methionine is going to act as a start codon. Because for that reason methionine alone, initially methionine will be converted into a modified methionine called as F-met. This is called as formyl methionine. So it is converted into F-met and then it is going to be activated. So first methionine alone will be converted into F-met, that is a modified form, and then it is getting activated. This is only when AUG is acting as a start codon. If suppose AUG is present somewhere in between the mRNA molecule, there a normal methionine will be added to the growing polypeptide chain. We'll see it, we'll take an example of that and we'll see it also. The second process is a transfer of activated amino acid to its specific tRNA molecule. This process is called as charging of tRNA. So in charging of tRNA is going to react with the amino acyl complex. So this is the amino acyl complex. tRNA is going to react with it, and then finally what happens is the amino acid gets attached to the tRNA and the AMP molecule and the enzyme is released from the amino acyl complex. So now this tRNA molecule which is going to carry the amino acid is called as the charged tRNA. Process is called as charging, and the tRNA that is obtained after charging process is called as the charged tRNA, and the tRNA has the amino acid in. So the amino acid is in the 3' end of tRNA. So attachment எந்த இடத்துல நடக்கும் அப்படின்னா it is going to be in the 3' end. So this is the second step in the protein synthesis process. Third step is assembly of ribosomes. நம்ம assembly of ribosomes நம்ம already பார்த்திருக்கிறோம். So when mRNA attaches to smaller subunit, larger and smaller subunits will assemble in presence of 0.001 molar Mg2 plus ions. So this is assembly of the ribosomes. The next step is going to be called as the polypeptide synthesis. So this is the actual translation process during which a polypeptide chain is formed. So to be clear with the process, we will first consider a stretch of mRNA molecule. இந்த mRNA தான் வந்துட்டு translation processல வந்துட்டு participate பண்ண போகுது. So it is going to be AUG, GUA, ACU, AAA and UAA. So this is going to be the 5' end of the mRNA. This is going to be the 3' end. 3' end, we know there is going to be a methylated guanine here and a poly A tail here. So this is the mRNA molecule. So now this mRNA molecule is going to enter into the translation process. So first வந்துட்டு இந்த mRNA molecule எங்க attach ஆகும் அப்படின்னா smaller subunit கூட. So we'll consider this is the smaller subunit of mRNA. And the larger subunit, we'll just represent it with bigger circle so that it is understandable for us. So this is going to be the larger subunit. So the larger subunit is going to have 3 sites, A, P and E sites. So this is the ribosomes that are assembled. So இப்போ வந்துட்டு இந்த mRNA molecule வந்துட்டு is going to get attached to this smaller subunit. So smaller subunit la vandu it is going to get attached only in one of its end. So this is going to be na inge oru line madri podren liya, this is going to represent the mRNA molecule. So in detail we will study nama idhe madri diagram represent panni vandu we will study. And one more thing, tRNA I am going to represent it only with a single line and an anticodon loop. ஏன்னா anticodon loop மட்டும்தான் நமக்கு வேணும், நம்ம full tRNA-வை வரைஞ்சிட்டு இருக்க வேணாம். Anticodon loop with the amino acid. This is how I am going to represent a tRNA, an activated or a charged tRNA molecule வந்துட்டு நான் இந்த மாதிரி மட்டும் represent பண்ணுறேன் diagramல. So first step வந்துட்டு என்ன அப்படின்னா, இப்போ smaller subunit இருக்குது. Smaller subunit identifies the the mRNA and binds to it. இப்போ larger subunitல வந்துட்டு நம்ம 3 சைட் படிச்சோம் இல்லையா, A, P and E site அப்படின்னு. இதில் வந்துட்டு more specifically start codon, that is AUG, that is closer to the 5' end of the mRNA. So நான் இங்கே எழுதிடுறேன் சைடில், the start codon will be identified only by the P site, and all other codons in the complete mRNA is going to be identified by the A site. ஸோ எம்ஆர்என்ஏவில் வந்துட்டு ஃபஸ்ட்டு பி சைட் வந்துட்டு ஸ்டார்ட் கோடானை மட்டும் ஐடென்டிஃபை பண்ணும். த ரிமைனிங் சைட்ஸ் வந்துட்டு வில் பி ஐடென்டிஃபைட் பை தி ஏ சைட். ஸோ நம்ம கன்வீனியன்ஸ்க்காக வந்துட்டு நம்ம என்ன பண்ண போகிறோம் அப்படின்னா, we are going to draw the mRNA like this. So this is going to represent the mRNA. So first 3. So this is going to be AUG. So this is going to be the codon number 1. This is the second codon, third, fourth and the fifth codon. So this is how the codons are present. நம்ம 3 lines மட்டும் represent பண்ணிக்கலாம், confusion வேண்டாம். So first codon P site வந்து தான் start codon identify பண்ண போகுது. So start codon is going to be AUG. So once P site வந்துட்டு start codon identify பண்ணிடுச்சு அப்படின்னா, immediately the tRNA that is going to have the anticodon for AUG. So AUG க்கு எது anticodon இருக்க முடியும்? For A it is U, for U it is A, and for G it is C. So this is going to be the tRNA that is going to carry. So we know methionine வந்துட்டு when it is to be carried for the start codon, it is going to be F methionine, that is formyl methionine. So this tRNA would be attracted towards this complex. So now, so first step, you are going to have the larger and smaller subunits, mRNA attached to it. This is A, P and E sites. இங்கே start codon இருக்கு. இந்த start codon கூட இந்த tRNA, that is the tRNA that is carrying F met is going to be attached. So this is going to be the first step. So meantime A site வந்துட்டு it is going to identify the second codon. So this is going to be the second codon. Second codon கூட வந்து second codon identify பண்ணின உடனே வந்துட்டு the tRNA that is going to carry the amino acid specific for this codon. So the second codon is going to be, so first codon is AUG. AUG க்கு வந்துட்டு methionine amino acid which is in form of formyl methionine is getting attached. Second codon வந்துட்டு GUA. So GUA is the codon and the amino acid that is representing for GUA is going to be valine. நீங்கள் அந்த நம்ம ஒரு செக்கர் போர்டு எழுதணும் இல்லையா, அதை வச்சு நீங்கள் செக் பண்ணிக்கிங்க. GUA வந்துட்டு codes for the amino acid valine. Now charged tRNA that is going to carry valine will be attached to the A site. ஏன்னா ஃபஸ்ட்டு கோடானை வந்துட்டு ரீட் பண்ண போகிறது P site, அதை மற்ற எல்லா கோடான்ஸையும் ரீட் பண்ண போகிறது வந்துட்டு A site. ஸோ இமீடியட்லி இந்த அமினோ அசைல் சைட்டில் வந்துட்டு this tRNA that is going to carry valine amino acid is going to come and bind. So now what happens is, once there are 2 tRNA molecules here, this larger subunit is going to act as the peptidyl transferase enzyme and is going to form a bond, peptide bond between this F met and valine. So now what happens, this is going to carry F met and this is the second codon. This is going to carry valine and there is going to be a peptide bond formation between F met and valine. So this is the peptide bond that is formed with the help of the peptidyl transferase enzyme of the larger subunit. So this is the second step. Now a peptide bond is being formed. Now what happens is, so once the peptide bond is formed, this ribosome moves through the mRNA molecule. That is, நான் வந்துட்டு ஒரு assumption காமிக்கிறேன் உங்களுக்கு. If this was the ribosome, so first step வந்துட்டு இப்படி இருந்தது, that is first site was here. So now once வந்துட்டு இங்கே ஒரு peptide bond, இந்த peptide bond form ஆன உடனே இந்த ribosome வந்துட்டு it is going to move like this through the mRNA molecule. So what happens is, the mRNA molecule, it comes out like this. So the first site is going to move here. So this is how the ribosome is going to move. இந்த மாதிரி தான் வந்துட்டு it is going to move to the next sites. So now when the ribosome moves, This is called as ribosome translocation. So next step is going to be ribosome translocation. இப்போ ribosome translocate ஆகும்போது என்ன ஆகும் அப்படின்னா, what happens is the first codon, that is AUG codon இருக்கு இல்லையா, இந்த codon வந்துட்டு it reaches the P site, and the second codon, that is that was in A site previously, comes to P site, and third codon now reaches the A site. So there is going to be a tRNA here with FMET. There is going to be a tRNA here with valine, and there is a peptide bond between these two. And now once this A site has the next codon, immediately the tRNA that is specific for the third codon. Third codon வந்துட்டு ACU கான amino acid வந்துட்டு it is 309. So now tRNA with 309 amino acid will come and attach here. Now what happens is, so once translocation happens and இது வந்து சைமல்டேனியஸாக நடக்கிறது, அதாவது ரைபோசோம் இஸ் கோயிங் டு மூவ், A சைட் வந்துட்டு 309 tRNAவை வந்துட்டு அட்ராக்ட் பண்ண போகுது, அண்ட் இட் இஸ் கோயிங் டு பைண்ட். மீன் டைம் இந்த tRNA இருக்கு இல்லையா, அதாவது FMET-ஐ எடுத்துகிட்டு வந்த tRNA இருக்கு இல்லையா, that is going to exit out. So what happens, what is remaining back after ribosome translocation is it is going to more or less look like this. That is, there is a tRNA which is having valine and FMET, and there is going to be another tRNA here with 309, and the tRNA that was attached to this site has left out. So this tRNA that has actually brought in the FMET has left the FMET with this amino acid, and it is going to move off. Now what happens is, when this valine is present here in the P site and A site has another amino acid 309, there will be a peptide bond formation between these two amino acid, that is between the amino acid present in P site and the amino acid present in A site. There will be an peptide bond formation. So the third step is going to be ribosome translocation. Ribosome translocation க்கு அப்புறம் வந்துட்டு the fourth step is it is a simultaneous process, the exiting out of the tRNA from the first codon and formation of peptide bond between the second and the third amino acid. This is the fourth step. The next step, so this is what has happened in the fourth step, that is there is no more tRNA here, mRNA has reached the E site, and P site la வந்துட்டு valine-க்கும் the second, so this is the second amino acid, this is the third amino acid, இது ரெண்டுக்கும் நடுவுல வந்துட்டு there is a peptide bond formed, and again there is going to be a ribosome translocation. So there is again ribosome translocation. So once again, when there is a ribosome translocation, what happens is, so this is what is going to be present, that is P சைட்டில் இருந்தது E சைட்டுக்கு வரப்போகுது, E சைட்டில் இருந்தது வெளியில் போக போகுது, A சைட்டில் இருந்தது P சைட், that is as such ribosome has moved like this. So the next codon would be read by the A site. So the next codon we have written is AAA, and for that an tRNA that is going to carry the specific amino acid. The specific amino acid is going to be lysine, is brought here. So once it is brought here, what happens is a peptide bond would be formed between the amino acid that is present in the P site and A site. So amino acid வந்து டைரக்டாக அட்டாச் ஆகிருக்கிறது வந்துட்டு 309, இங்கே லைசின். இது ரெண்டுக்கும் நடுவில் வந்துட்டு there would be peptide bond formation. Meantime the the tRNA that has brought in valine is going to leave out. So what is going to remain back after the ribosome translocation? So it is going to have, see, it is going to be 309 attached to valine, valine attached to F-met, and there is the next codon here in the A site, and this is going to have lysine, and there is a peptide bond formation between 309 and lysine now, and the codon the tRNA that has actually brought valine has left out now. So this is happening after the ribosome translocation. And what happens is, once it has left out, the ribosome translocation is going to happen once more. So once more, when there is ribosome translocation, what was present in A site has moved to P site now. So lysine, it has moved to P site, and what was present in P site, that is the tRNA with 309, has moved to E site. So once E site போகும்போது 309 is attached to lysine. So இது இங்கே attach ஆயிருக்கும். இந்த tRNA வந்துட்டு it is going to exit out. The next codon comes to A site. Next codon, what we have written is UAA. The next codon what we have written is UAA, which is a stop codon. So once A site வந்துட்டு, if it is identifying stop codon, then so immediately, so now what happened is, what has happening here is going to be the A site identifies stop codon, that is UAA is identified by the A site. So immediately what happens is, release factor comes into action. So எப்போ ஸ்டாப் கோடான லார்ஜர் சப்யூனிட்டோட A சைட் ஐடென்டிஃபை பண்ணுதோ அப்போ வந்துட்டு ஒரு ரிலீஸ் ஃபேக்டர் அப்படிங்கிற சப்ஸ்டன்ஸ் வந்துட்டு will come into action here. So what is this release factor அப்படின்னா, release factor, it is commonly called as eukaryotic release factor 1, also called TB31. So this is the name of the release factor, and this release factor, what it will do is, it is going to, and it can recognize all 3 stop codons. So it is going to verify whether the A site has identified the stop codon or not. So it can recognize the stop codons. Once it recognizes the stop codons, what is it going to do is, it is going to release the polypeptide chain from the ribosomes. So once the release factor has come into action here, immediately the polypeptide chain is going to be released from the ribosome. So polypeptide chain அப்படிங்கிறது என்ன? It is a chain of amino acid that are linked with the peptide bonds. So the polypeptide chain that was formed here was this. This is the polypeptide chain that was formed. நம்ம ஒரு எக்ஸாம்பிளுக்காக ஒரு சின்ன லென்த் எடுத்துருக்கிறோம். It is going to be many amino acids length in case of many proteins. So this is going to be released out. So this is released out. So once released out, what happens to the remaining substances? So once this is released out, the ribosomes dissociate. That is, and the assembly வந்துட்டு dissociate item, larger and smaller subunits are going to be separated from each other. Then the mRNA is degraded by the enzymes of cytoplasm. So it is immediately degraded. So this is, so finally என்ன பேலன்ஸ் இருக்கும் அப்படின்னா இந்த ரிலீஸ் அவுட் ஆன இந்த பாலிபெப்டைட் செயின் மட்டும்தான் வந்துட்டு இங்கே இருக்கும். அப்போ வாட் ஹேப்பன்ஸ் டு திஸ் பாலிபெப்டைட் செயின்? This polypeptide chain that is usually formed after translation is going to be an inactive protein. So this polypeptide chain is going to be taken into Golgi apparatus and there it undergoes activation process and is secreted out by Golgi as activated or functional protein. So in the protein vandittu it can act itself as protein itself. It can be converted into some enzymes, structural proteins irkalam, functional proteins irkalam. So there are different types of proteins and the madri vandittu they are converted into whatever type it is needed and it is going to be released out by the Golgi after activation process. This activation process that is taking part in the Golgi apparatus, it is called as post translational modification. And one more thing we have to remember is the untranslated regions. Untranslated regions of mRNA அப்படின்னா நமக்கு சிம்பிளாக புரிஞ்சிடும். பிகாஸ் நம்ம ஸ்டார்ட் பண்ணினது எஃப்மெட், அதாவது ஸ்டார்ட் கோடானில் இருந்து எங்கே முடித்தோம் அப்படின்னா ஸ்டாப் கோடானில். ஸோ இப்போ ஸ்டார்ட் கோடான் டு ஸ்டாப் கோடான் வந்துட்டு எம்ஆர்என்ஏ மாலிகுல், ஃபங்க்ஷனல் எம்ஆர்என்ஏ. ஆனால் அதோட ப்ரொடெக்டிவ் பார்ட் அப்படின்னு ஒன்று இருக்குது, அதாவது பிஃபோர் ஸ்டார்ட் கோடான் அண்ட் ஆஃப்டர் ஸ்டாப் கோடான். அந்த போர்ஷன்ஸ் தான் வந்துட்டு we call it as the untranslated regions. So it is going to be untranslated regions are going to be present before start and after the stop codons. So these are also important for an mRNA molecule, but those are untranslated regions. So this is the process of translation, that is protein synthesis happening in eukaryotic cells. So we have completed translation process with this. We'll meet with the next video about regulation of gene expressions in prokaryotes and eukaryotes. Thank you, students.